Hi everybody, welcome to another Chats with Children. I hope you're safe and well. Today I'm delighted to be joined by Gary Nabel, who is the president, CEO and co-founder of ModeX Therapeutics. And the reason I'm speaking to Gary today is that he is a keynote speaker at PEG's Boston next week, which obviously is not in Boston, it's virtual. And he is doing a keynote presentation on tackling AIDS and cancer with trispecific antibodies. So Gary, thank you for joining me at such short notice.
It's my pleasure. Thank you.
Well, before we start talking about your presentation and what people can expect to see when they listen to you or hear you, perhaps you give people a little bit of a background on ModeX if they're not familiar with the business, and also your background.
Well, why don't we start with ModeX? We started as a standalone company in November of last year.
Right.
And the main thrust of the company is really to develop multi-specific antibodies. We are looking at this point to develop new ways to generate multi-specific antibodies, and also to support the development of prototypes that we developed in the past. Many of them, and in fact, much of the start of this technology came from work that was done by a team that I led at Sanofi,
Right.
where I was the chief scientific officer. A large part of that team has come to ModeX, and we hope to grow the technology and really apply it to places where it can help patients in the future. That's what we're all about.
Fantastic. And given it's such a relatively new business, how's business been going, and what's the feedback been from the industry so far?
Well, we're still in what I call stealth mode. We're trying to put together our own programs and build a base of both knowledge and science and medicine and financing to get launched. But it's really, very exciting to be in that environment. We're small, but we're nimble, and it's refreshing after many years, both in private sector and in public sector, being parts of large organizations. We can do things much more quickly.
Sure. It's scary, but it's also exciting at the same time, isn't it?
Yeah, absolutely.
Fantastic. Well, let's talk about your presentation next week. So I'll ask a very basic question because I'm not a scientist. So what is a trispecific antibody?
Well, a trispecific antibody is one that we've engineered in the laboratory. It doesn't occur like this in nature. Standard antibody is Y-shaped. It has two arms and
Sure.
it has a base. And at the tips of the Y, both arms engage the same target. So it's monospecific.
Right.
A trispecific antibody has been engineered so that there's a double stack of antibodies on one side, and they
Right.
each recognize two different things, and they're paired on the other side with yet a third antibody recognition site. So we essentially pack all of that information into one molecule that normally would have been spread out between three.
Right. Okay. So that's partly answered, I think, my next question. So what are the advantages to using a trispecific antibody compared to a standard antibody?
Well, there are several. Our first work in this area was published in Science in 2017, and I'll talk about it in my keynote talk. And there's one example where three is better than one, because with HIV, although we are able to identify single antibodies that can neutralize HIV, any one of those single antibodies, you can generate resistant mutants of virus. And so you are still saddled with the problem of escape from immune control. By having three in one place, the virus now has to essentially change three parts at once in order to escape neutralization. And that's very hard to do. So that those additional specificities give you more breadth. They also give you more potency, because you're able to interact with more of the components of the virus at once. And so those are what I would call the standard aspects. You can recognize different things at once, and they're more potent. And that has a lot of value in cancer research, in AIDS research. I think it's very applicable to COVID as well. The other thing that it allows you to do is that, particularly in cancer immunotherapy, it allows you, when you can target different things at once, it allows you to bring things together that normally don't come together. And so, for example, if one of the arms of the antibody is directed to a tumor and the other is directed to an immune cell like a T lymphocyte, you can actually bring them together, deliver signals to turn them on. So it's a very interesting way of co-locating molecules and doing it all in one compact unit.
Fantastic. But what are the limitations, though?
Well, the limitations at this point really relate more to the unknowns. So, for example, we know that it's very standard to manufacture normal antibodies. You can get very decent yields and they can be very cost effective. At the beginning, it really took us a while to convince ourselves that they could be manufactured at scale. And so there are issues related to just the scale up and the technical aspects of putting them together. There are challenges when you put three things together. It's not always, what's nice about the technology is that it's what I call plug and play. It's like putting cassette tape in, but there are different ways to put the units together. And so you have to find out how to best configure it, what's most manufacturable, and also make sure that you don't introduce any red flags to the immune system so that the immune system says, oh, I don't want you here, and gets rid of it.
Right. And you've done it with three. So is it possible to make antibodies with more than three specificities? And if so, how would and where would they be used?
It is possible, in fact, there are in fact different technologies for trispecific antibodies. We use a standard antibody format where we stack things one on top of another. So you can imagine if we have a double stack on one side, we could put a double stack on another side, and it would be pretty easy to make four. And we have made four at times, so that's not a big deal. Where there are other technologies, for example, some of the camelid antibodies, they're like beads on a string. You just line them up and link them together, but they're not connected to standard antibody frameworks like the FC binding region. But again, I think there are multiple ways in which that technology could be useful, and I think there are ways where some of the hurdles with the higher valencies could be worked out. But just as another example, in cancer cells, we actually do much better identifying cancer cells when we look at combinations of proteins on the surface rather than single proteins on their surface. So we can get more specificity by targeting two things at once. And then if we want to bring in, for example, lymphocytes and activate them, we can use the other two arms for that. Similarly for viruses, if you make it so that the virus has to mutate against 4 different determinants, that's an even a higher mountain for the virus to climb. And so it can make your antiviral therapies more effective. So I think it is an evolving technology with a lot of different applications. And it's just a matter of figuring out where they can do the most good and how to make the best ones.
Brilliant. Well, thank you for doing that. I forgot to ask you, because I'm looking at the trees and the sun out behind you. Where are you calling it from, by the way?
You wouldn't think I'm in Massachusetts, would you? I'm not. I'm in Florida, actually. But
all right, I was going to say, with the sun shining. Well, there you go. I should have worn my Dolphins jersey then if I knew I was going to interview in Florida. So, well, Gary, that's been really, really interesting. Thank you very much for taking the time out to sort of give a quick overview of the subject. Obviously, if people would like to know more, they can, and they're attending PEGS Boston next week, they can check out your presentation, and I'll put details of the time and date of that presentation above the video so people can see that and click through to it as well and register for that particular talk. But I really appreciate taking time out, and it's very short notice. I mean, literally, because we arranged this in the last half an hour, this interview, so I really appreciate Gary taking the time out to do this at short notice.
Yeah, enjoy your weekend. And by the way, there will also be a panel discussion. So I think if people have questions, they can tune into the panel discussion as well. So it all sounds good.
Fantastic. Well, I wish you lots of success next week. I hope it goes really well for you, the presentation, and also lots of success with the business as well. You know, hopefully you'll come out of stealth mode, and I'll be hearing and reporting on you, hopefully on LinkedIn, all the great discoveries that you do. So thank you for taking the time to talk to me, Gary. Good luck, and thank you very much. And thank you everybody for watching. I hope you found that useful. If you are at PEGS Boston next week, make sure to check out Gary's keynote presentation. And if you're not, the link will be up there to register as well if you'd like to know more anyway. And Gary, one final question actually. Where can people, if they can't make the presentation, get more information about your technology?
We do have a website. It's pretty nascent, but you know what I would really suggest is in my talk, I've cited a few papers, and those papers do a pretty nice job of covering the landscape, and that's a good place to start.
Brilliant. Well, what I'll say then is maybe if you could send me those links, I'll put them in the video above the video as well, so people can see those links. That would be great. So thank you very much once again. Look after to have a great event, and thank you very much viewers for watching. Check out the presentation next week, look at the links, and actually if you've got any questions, Gary, and you're not at the PEGS, put them in the comments below the video. I'm sure Gary will be happy to answer them. And until next time, as always, stay well and stay safe. Bye-bye.